Hi and welcome. Today we will discuss the features and structure of DNA found by scientists Watson and Crick. Here we can see DNA. DNA is made of subunits called nucleotides that are arranged to produce the double helix structure of DNA. Nucleotides consist of a sugar nitrogenous base and a phosphate group. In DNA, we label the nucleotides by the ribose carbons. Carbon 1 is the carbon directly attached to the nitrogenous base. Carbon 2 distinguishes DNA from RNA, where DNA will have an H group attached, and an RNA would have an OH group attached. The 3' carbon is bonded to the phosphate group of the next nucleotide. Lastly, 5' is attached to the phosphate group. The connection between the 3' and 5' carbons are what form the sugar-phosphate backbone, which are held together by phosphodiester bonds. There are 2 strands that consist of nitrogen bases that bind and hold together the DNA. These nitrogen bases are separated into 2 groups, purines and pyrimidines. The purines are adenine and guanine, which are double ring structures, while the pyrimidines are cytosine and thymine single ring structures. These bind A to T and G to C, as seen here. They are held together by hydrogen bonds, which are relatively weak and do not stabilize the DNA double helix. Strong hydrophobic interactions in the base pairs are what keep them together. This is maximized through base pair stacking and the interactions. This is found between the bases which stabilize the DNA. DNA is also arranged with one strand going from the 3' end to the 5' end, and the second going from the 5' end to the 3' end. This is called anti-parallel. DNA also has handedness, which is determined by the direction of the helix. DNA is mostly right-handed, as seen on the left. And how we can figure this out is by starting from the bottom of the helix and acting like we're walking up the staircase. If we start with this one on the right, you're going to put your hand on the blue side, which is your right hand, and walk up. If we looked at the left-handed helix, we would start at the bottom and use your left hand to grab onto the blue side and walk up. There are also major and minor grooves that show up in DNA strands. More base pairs are able to interact in the major grooves due to more space. Lastly, let's look at melting point. One of the destabilizing forces of double-stranded DNA are the negative charges that are present in the phosphate backbone. When DNA molecules are placed in a higher concentration of salt solution, then they partially neutralize that negative charge. So, negative would be partially neutralized if added to a salt concentration buffer because the DNA would be held together more tightly. So the amount of heat required to break apart or denature the molecule would go up. The point at which 50% of DNA is denatured is the TM. So the TM would go up if mixed in a higher salt concentration buffer. Thank you for watching.